So I can't remember, do the T cells live a long time, or do we just end up with less of them?
You end up with less of them, and when it comes to survival, that's kind of a dual-edged sword. They only survive for a handful of days technically when they become activated. When they're not actively doing something, they're referred to as kind of pre-T cells, which is non-activated T cell. They only become active when they get exposed to a foreign invader. So a foreign antigen gets in the lymphatic fluid or gets in the blood, and this T cell happens to be there, then it sees it, then it will mount, well, then it differentiates, become active, and then it starts to mount an immune response. And again, they only last for maybe, well, the immune response is about 14 days when you first get exposed to something. So those T cells will only last for a handful of days. And then they call more into the party. And then if they can't kill it off, they'll call in the B cells that we'll see here and mount an antibody immune response. So T cells only last for a handful of days to begin with when they become activated. Prior to that, I've never seen a specific shelf life of them. We have a few different types of T cells, and we have memory, and memory lasts forever. So those type of T cells can hang on forever. There's just no space for them in the thymus anymore, so they atrophy and decline in numbers because there's not enough spaces within the thymus to hang out and be cool. Our secondary lymph structures, everything that's not blood bone marrow or red bone marrow, sorry, or the thymus. All right. So these are going to be spleen and lymph nodes. Those are the kind of the big ones. All right. These guys have a defined capsule. What does that mean? That means they are there. You can see them individually. All right. If they don't have a complete capsule, then they're not completely walled off from everything around it. So they can kind of get lost in a structure. Tonsils have a little bit of a capsule and don't have some capsule spots. Malt or diffuse lymphatic nodules, again, malt doesn't really have a capsule completely around it, so it's kind of within the tissue there. Malt is mucus-associated lymphoid tissue. We have salt, skin-associated lymphoid tissue. We have galt, gut-associated lymphoid tissue. So we have a lot of this diffuse tissue all over the body. Okay. And it's kind of interesting where we see those, and hopefully, it will make sense when we start to talk about those. All right. But our spleen and our lymph nodes, these are, when we think of secondary lymphatic structures, these are the two that we really think about. All right. The spleen, just because of where it's at and what it does, its major function is lymphatic system in nature. And lymph nodes, good God, if you've ever been to the doctor and you've been sick, your doctor's palpated your lymph nodes and you've heard the word, you understand what they are. They're definable, tiny little round structures that you can palpate and feel. So these have complete capsules around them. They house lymphocytes. These house lymphatic cells. They are not where they are formed, though. They hang out. And when they get exposed to somebody, then they differentiate and attack. So here's what a typical lymph node looks like. Okay. We have a number of vessels, afferent vessels, that bring in lymphatic fluid. These are lymphatic vessels, right? Okay. We have a number of those. As you can kind of see, a number of them can go in there and feed into a individual lymph node. Okay, so we have many of these, many. Okay. We have this little indentation here, all right? This indentation, guys, is referred to as a hilum. So everything for the most part is going to have a cortex outer and a medulla inner from here on out. Another structure that we're going to see in a lot of different things from here on out is called the hilum. And the hilum is this indentation region right here within a structure. And the hilum is where we have one efferent vessel leave. Many afferents, only one efferent. Can anybody think of a reason as to why that would be significant or what that would cause to happen here? Multiple afferents and only one efferent. Can anybody think of what that's going to cause?
Change in pressure?
Yeah, right? Would this cause a backup? Well, quote unquote backup? Yeah, right? If I have 5 cars entering at one time, but only one exit, well, now they can only go out one at a time. So what does this many afferent, one efferent vessel establish? It establishes a slowing of fluids through the node. This ensures that the T cells and B cells that are housed within the cortex and the medulla get exposed to that foreign invader. It slows lymphatic fluid movement through the system, just a little bit. And it's just that little bit of slowing it down that can allow that B cell and T cell to be exposed to that foreign invader and mount an immune response. They're small, they're ovalish, blah, 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 okay? You see them superficially, which, I mean, palpate and feel, okay? They're found deep. They ride along a lymphatic vessel. They can be clustered in spots. Right here. You have some cervical lymph nodes here. You have some cervical lymph nodes in the back. They're all clustered up there. Whenever you have this on the web. Sorry, gosh. Whenever you have a infection, whenever you have a cold, a head cold, why is it always, just you're swollen and inflamed, you're fighting stuff off, your neck is so swollen and sore to move? It's these lymphatic vessels, these lymphatic nodes that are super swollen and slowing movement of lymph through the system. We have some in the groin. Okay, it's why you may feel really stiff in your groin or the popliteal region behind the knee. In the armpit, you have a huge number of them. Okay, here in the armpit, here you have an anterior compartment, kind of right behind that pec muscle. You have a medial portion right along the wall of your thoracic cage. You have posterior right there on that subclavian muscle. And then right here on the arm here, okay? And that's really sensitive right there, okay? So that axillary lymph node is doing a lot of work there, checking the fluid as it moves through the systems. So you'll find them a lot of cluster in a lot of different spots. It has what's called trabeculae, and this is just what compartmentalizes this lymphoid structure, okay? kind of helps divide into a outer region here called the cortex and an inner region called the medulla. Okay. And what's in that cortex and what's in that medulla? Well, lymphocytes. Okay. In the cortex, we have what are called cortical centers. Okay. Germinal center in this book. Here is a germinal center here. Germinal center. Okay. Germinal center. And it's within this germinal center where we find a number of lymphocytes. T lymphocytes. They'll hang out here. They're called a pre-T lymphocyte, but I just have T lymphocyte. Okay. They just hang out here, waiting for somebody to bring them a foreign invader. We have B lymphocytes. B lymphocytes make antibodies. They'll just sit here, be hanging out within this germinal center, get exposed to a foreign invader, and they'll start differentiating. Did you see it for the first time? Did you see it for a second or multiple time? Well, that will depend on the type of B lymphocyte that you form. Is it bacteria? Is it virus? Is it parasite? That will dictate the type of B lymphocyte that you form. We also have a number of just lymphatic cells, or immune type of cells, I guess I should say. Macrophage, general chewer-upper of stuff. Okay. If something's in the body that should not be there, macrophages are generally just circulating around in the blood and in and out of tissues, and they'll just come up and swallow it up. Try to destroy it. If it can't, we'll see here in a little bit that macrophages actually take this foreign invader to a T lymphocyte. Sorry. Dendritic cells are also another general type of immune type of cell. Dendritic cell you find mostly in skin and tissues like that, epithelial tissues. So if something gets through your skin, which is a barrier or a boundary, let's say you cut your skin or your skin gets cracked because it's dry for some reason, foreign invaders can get in, dendritic cells will mount an immune response. Okay. And they'll bring it to a T cell. So the cortex contains these germinal centers, and it's these germinal centers that house a number of lymphocytes, whether it be B or T. Deeper within this lymph node, we have what's called a medullary cord. Here we're going to have a lot of T cells, B cells, just hanging out, getting ready to differentiate. And that's it. When you look at this structure, we have a number of afferent vessels and only one efferent vessel. A lot of fluid coming in, but it backs up a little bit, so it doesn't quickly move through it. Many tributaries coming in, but only one leaving creates a little backlog. And that's enough to help to slow fluid movement through this thing. So these B cells and T cells, macrophages or dendritic cells, can see the foreign invader, differentiate, and mount an immune response. We also have a number of sinuses, and I didn't get into it. There's a subcapsular sinus, there's a medullary sinus. There are a number of sinuses, and sinuses are just a little bit of space that exist within this structure. And if sinuses are a little bitty space or a little bitty void, that void would need to be filled, and then fluid could then continue to percolate through it. So these medullary sinuses will also slow the movement of lymphatic fluid through the system as well. Okay. So not only do we have a handful of vessels coming in and only one leaving, slowing it down, we also have a few different sinuses doing it as well. And all of this is to do one major thing, and that is to slow the movement of lymphatic fluid through this system. And that's kind of basically what I said there. Okay. Along the length of a lymphatic vessel, guys, it is important to remember that there's going to be a number of these little lymph nodes scattered along it. Okay. So will it be able to slow fluid enough down to get those T cells or B cells to differentiate and mount an immune response? No. But what's it doing? It is priming the system. And as soon as this starts to get through more and more of these lymph nodes, you're going to have a proliferation and an increase of B cells and T cells within the lymphatic fluid, and slowly you mount an immune response. It takes a few days to get this to happen, all right? Especially if it's the first time you're seeing it. Don't worry about the slide. I didn't ask any questions about it, so I'm just going to move on. Nothing about non-Hodgkin's or Hodgkin's lymphoma. Okay. Our second major secondary structure is your spleen. And the spleen is unique. And because it is unique, there'll be a handful of questions dealing with that. It is very superficial. It is in this posterior lateral region there on the left side that is very exposed, okay? It is not protected by a whole heck of a lot. And because it is not protected by a whole heck of a lot, it can be easily punctured and easily damaged. And it has a highly extensive vasculature network inside of it. And because it has so many blood vessels, if you do puncture it, you got to get it removed usually right away, because you'll bleed out. It is that much. It has a hilum. Again, I don't think I asked any questions about the term hilum. It's just something that you have to be comfortable with. Hilum is generally where multiple things enter and exit a specific organ, as we'll see from here on out in the rest of the semester. It has trabeculae, helps to compartmentalize it. It is its histology that makes it unique. It is its histology that makes it unique. It has a couple of different clumps of tissue within it. We call this tissue white pulp and red pulp. And if you just know that erythrocytes are red blood cells and leukocytes are white blood cells, then you can kind of get a general gist as to what these two tissue types are predominantly associated with. White pulp is associated with immune response. Red pulp is associated with recycling old defective red blood cells. Okay. Every 120 days, remember, that spleen helps to recycle these, break these things down. It's a storage site for some of the parts and pieces that go into making a red blood cell. And that's what primarily red pulp does. All right. As you can kind of see here, the white pulp there and the red pulp there on that histology of the spleen picture right here, you can see the white pulp looks whitish and the red pulp looks reddish because it's perfused with blood. The white pulp not as perfused with blood. So, I mean, there's the functions there. Again, it's probably a multiple choice type question. It's not that difficult. Most everybody generally gets it right. It's a freebie. Yeah, pretty easy. Something that's unique about it, in the first 5 months of life, of fetal life, the spleen makes all the blood cells. So that's kind of interesting there, fetal life. So that's kind of interesting there. This can be reactivated under certain conditions, certain hematological conditions. So the spleen does a lot of heavy lifting in the very beginnings of life. And then that kind of goes away unless certain kind of situations manifest. But it monitors the fluid, monitors the blood, so it can help mount an immune response. You can remove the spleen. It does recycle old defective red blood cells. It is a reservoir. But I mean, you can get the spleen removed and the liver will do that. Okay. And that puts a little bit more stress and strain on the liver, but the liver can do it. Okay. White pulp, though, I mean, that function's lost. The liver can't do that one. Okay. The liver can't do that one. So your immune response is a little bit dampered if you have to have your spleen removed, which would be a bad thing. Tonsils and MALT, lymphatic nodules, are other secondary structures. What would I ask about these things? Where are they located? We have pharyngeal tonsils up there in the nasopharynx. Pharynx is right here. It's this shared tubular structure between the respiratory system and the digestive system. That's what the pharynx is. But you breathe through your nose, right? So a foreign invader can be inhaled. Pharyngeal tonsil is going to see it. You can drink. You can eat. You can mouth breathe. And if we have a foreign invader come in that way, we have a palatine tonsil and a lingual tonsil that can be exposed to this thing. What is this? This is an opening to the outside world. Any place where we have an opening to the outside world, we are going to find a large number of lymphatic tissue. And up here, we have the nasal and oral cavities covered with our different tonsils. Your adenoids those are your pharyngeal tonsils. Okay. When they become enlarged and inflamed, that's when a pharyngeal tonsil gets called an adenoid. Okay. Palatine tonsil is in the upper portion of the oral cavity, and the lingual tonsil is in kind of the basement of the oral cavity. But we have our entire openings covered. So if you breathe it in or inhale it in, or chew it up, swallow it in, you are covered. When you look at these things, these tonsils are unique. Okay? They are there for immune surveillance of anything inhaled or ingested, like I just said. They have what are called crypts. Okay? And when you look at it on a side view, a little bit better one, okay? Let's make it really simplistic. It contains this tonsillar crypt. Okay? Think of it like a grave. Bacteria can come on down here and get trapped, and we can mount an immune response. If for whatever reason you have chronic infections, it can cause, that bacteria or whatever is getting in there. can cause that lymph node to be excessively swollen, and that's when you'd have to have them removed potentially. So it is the gift and the curse that is a tonsillar crypt. All right. It's where foreign invaders can get trapped and you can mount an immune response. We have germinal centers within it. So we're going to have B cells and T cells within here. So that's how we immune surveil. Okay. We monitor what we are breathing and ingesting in. Aside from those tiny little structures, we also have some diffuse tissue. Okay. And this diffuse tissue is found pretty much everywhere within the body, but it's going to be concentrated in areas that are open to the outside world. Why would it be concentrated in areas that are open to the outside world? That's where the foreign invaders are coming from. So even though they're going to be concentrated in the oral cavity, in the anal cavity, in the urethra, and anything that's open to the outside world, they are still found in most every body organ. It's just like a lymph node, except it's non-capsulated. That's all it is. Okay. It's diffuse. It's dissolved within the tissue. That's what that term kind of means. The most important of this diffuse tissue is MALT. And MALT stands for, makes a good question, mucus-associated lymphoid tissue. What does the GI, the respiratory, the genital, and the urinary tracts all share? What do they all share, guys?
Malt.
Ha. I love you. Yes. What else do they all share? They're all open to the outside world. They're all open to the outside world. Every single one of those has an opening to the outside world. Mouth, nose, anus, all of them. External urethral orifice, all of them have an opening to the outside world. That's how things can get in. So you're going to find them really in the lamina propria of all of those regions there. In the small intestine, we have a very important MALT type tissue. The small intestine consists of the duodenum, the jejunum, and the ileum, 3 different portions associated with this structure. And in that last little bit of the small intestine is where we find Peyer's patches. These are some specialized lymphatic nodules that are only found here within the small intestine. 2 things. 1, we have the anal opening. We can have a foreign invader come up there through the back door. Sometimes foreign invaders get activated after being exposed to the acid contents of the stomach. So if that's the case, once they become activated, well, now I have Mr. Peyer patches there, and it can see this foreign invader and mount an immune response.